Hello, my name is Lurdes Duarte. I'm the R&D director at Isogenica, and today I'm going to be talking to you about our fully in vitro display system, CIS Display. Let me start by giving you a very brief overview of Isogenica. We are the VHH company. We are located in Cambridge, UK, at the very nice grounds of the Chesterford Research Park. We have over 25 years of antibody discovery experience. We've delivered for a number of partners, and that we know of, we have 3 candidates that have progressed into clinical trials. And at any time when you talk to us, we will have several discovery projects running in parallel. So today I'm talking to you about display systems. As you may well be aware of, there are several display technologies that are very well known. Some of them, like for example, yeast and mammalian display, are really good for complex scaffolds, like is the case of SCFEs, but they will struggle if you are trying to display large libraries. In which case, we would move to technologies like phage display, RNA display, or CIS display. At Isogenica, we do perform phage and CIS display. Phage display is very well established. We use it on a regular basis, and we have very successfully delivered with this technology. But we also have the largest VHH libraries in the market, in which case technologies like CIS display and RNA display will be ideal so that we can make the full use of our libraries. RNA display, while it is very good at displaying large libraries, is not the most efficient or stable of the display technologies. Comes in CIS display, our funding technology. It's a DNA-based cell-free display, which is excellent at displaying large VHH libraries. So as I mentioned, CIS display is a cell-free DNA-based system. Because it's cell-free, it does not require a cell transformation step, which is usually the limiting factor to the full display of the libraries. It has been validated in different scaffolds, VHH, peptides, centurines, and while we use it with our large libraries, it can also be used with a library of your own. So because it's scalable, it's very suitable for high data output. A bit more detail into the technology of CIS display, the biology behind it. As I mentioned, is a DNA system. So we start with a linear double-stranded DNA template that contains a library and is linked to the RepA-CIS-ORI region. Then we incubate this double-stranded DNA with a bacterial lysate, and this is what we call the ITT reaction, which stands for transcription and translation in vitro. So the transcription process will stall and terminate at the CIS region, and this allows for the RepA to find its own DNA-specific ORI binding region. So the peptide being displayed is linked to the very specific DNA from which it was transcribed. What this means is that the proteins that you are displaying are specifically linked to their own DNA, so phenotype and genotype are linked, which is what you need in a good display technology. This has been demonstrated experimentally with peptides, so we have papers on that. If you are interested, please go and explore more details on this technology and check the outcome of its use. Now, because of its scalability, this is a technology that is very suited to complex projects and in silico informed triage. It is cell-free and it can be usually done using 96 well plates. So we know that we can do at least up to 48 different conditions at the same time. So each round of selection takes 1 day. If you have a complex project where you are not 100% sure of what would be the right conditions to get the VHH with the ideal characteristics to your mode of action, this is perfect because you can test all of them, different combinations at the same time. Is the epitope what is really important for the success of your project? Are there different epitopes that you want to mask that probably will direct the selection towards the epitope that you desire? Do you need your VHH to behave differently under different pH conditions? How many species cross-reactivity do you need? So we can interrogate for all of them at the same time and include also negative streams that will allow you to subtract the hits one from another. The beauty of this is that you can perform NGS analysis in each one of these wells. So not only you can do the empirical testing to see where the desired hits come from, you can also do in silico informed triage using these negative selection streams for each of your positive conditions. And what this allows is to identify additional binders to the ones you will be identifying empirically. So it will increase the diversity of the hits that you get out of these selections. One additional thing that this system renders itself to is to train your ML models and to optimize AI libraries that you may have generated. So as I mentioned initially, we certainly can include our large libraries into this system, but we can also include a library that you have designed. So we have done this, for example, for centurines that were very successful. If you have VHH or other small scaffold libraries that you want to test and improve, this is a very good system to do that. Because it's cell-free, whatever data that comes out of it is very clean. As I said, we can interrogate many streams at the same time. So while you can design the right conditions to get hits against your target, you can also include which conditions do you need to inform your model to further optimize the design of your library. And this is a very quick system. You can do all your round of selections in 1 week. So you will have that data, feed it into your model, redesign, retest. It would be very straightforward progress. And this is maybe a limiting view of how our CIS Display system can support ML training and AI design of new libraries. So if you have additional ideas on how this flexible and fast and clean system can help your project, just come and talk to us. We'd love to hear from you. In the papers that I mentioned in the beginning of my presentation, you can find examples of successful discovery campaigns with peptides using CIS Display. Here I have a case study of discovery campaigns using CIS Display with VHH libraries. So we did do a case study to select hits against PDL1. So I chose just to show a few of the top hits that we got from these selections, but I think it's enough to give you a flavor of what you can get out of our libraries together with this system. We have used as a positive control durvalumab Fab, and one test that we did was to check for the ability of our VHHs to inhibit the PD1-PDL1 interaction. As you can see from this example, we do have the durvalumab Fab doing 100% inhibition with very good affinities. And from our 4 hits, 3 of them do inhibit 100% with different characteristics, and one of them there's only partial inhibition. So this indicates that there is diversity in the pool of primary hits that we get. Well, this is quite relevant. Something that is key is there a translation between what you see with recombinant protein and what you see then with cells? We did test these 4 leads for cell binding, and as you can see, they all bind equally well to cells. So EC50s to recombinant protein, EC50s to cell lines. We have 3 libraries. 2 of them are humanized, but 1 of them is still on the llama background. So the hits that I'm showing you here actually come from the llama library. Something that we can also do at Isogenica is to humanize these llama VHHs, because there's a fixed framework scaffolds. We know very well what changes can be made without affecting the functionality of these hits. And this is what you can see here. So for example, for clones 1 and 36, this asterisk means that when we measured KD for human and cyno, actually this was done in the humanized version of the primary hits. And as you can see, they do retain very good binding ability to the target. Developability is a very important characteristic that we want to know as early as possible. So for these clones, we did check for thermal stability and percent monomer. And as you can see, we have very good values in terms of thermal stability, and the same is seen in terms of percent monomer. In the cases where this value is lower, this is due to the low scale production to which this analysis corresponds. For the example of clone number 4, when we performed large scale production, we did observe a much better percent monomer, so over 99%, which is excellent. So good developability conditions. Something that is very likely to happen in your project is that you are not intending to use the VHH as a monomer as its final application. Probably you are looking to do ADCs, CAR-Ts, bispecific formatting to whatever additional molecule. So it is important to also understand, do these VHHs accept formatting? We did do that testing. We have linked our VHHs with a T-cell engager, which in this case is an SCFV, to form bispecifics. We also did trispecific formatting, in which we include the albumin binding VHH and ISO extend in case extension of the half-life is something that you desire. Again, we used durvalumab as our positive control, and we tested this in killing of PD-L1 positive cancer cells. As you can see, durvalumab has an excellent killing capacity, but so do our bi or trispecifics. So formatting did not affect their binding abilities and resulted in a low picomolar efficiency in killing. So these VHHs come directly from our library. No engineering, no optimization was done to it. So the potential is huge. Just a summary of what I've presented to you and some additional notes. So as the VHH company, our core work is to do early discovery using our synthetic large libraries. CIS Display has an immense potential, but phage display is also something that we perform routinely and very successfully. As I've shown you, we can humanize it if they come from the Llama library. We have a very customized service. It will all depend on what you are looking for. There's a conversation, we listen to you, we explain you how things can work, what's the best route to success, and then we go from there. We always do thorough antigen QC to guarantee that what goes in is as best as it can. And we can do high throughput screening and NGS analysis for you. We can also do VHH characterization, as I showed you. We have rapid high throughput expression, and we can do biophysical profiling. We can test affinity, epitope binning, specificity, species cross-reactivity, and in addition to that, we can also do some costume functional assays. I've showed you, for example, the cell killing assay. We can also do internalization, antagonism, other things that you may need. We can work with primary human cells, and in case we do not have the ability or there still needs development for the assay that you are looking for, we have partnered with Axam and Charles River, which can take you further down into IND. And because all the legal paperwork is sorted, it's a straightforward process if this is something that you are looking for. I also showed you that we can do antibody engineering. So one example was the bispecifics, humanization. We can also do affinity maturation, and we can add additional tags and payload handle integration. Okay, so thank you for listening. I hope I gave you a good idea of what can be achieved with our large VHH synthetic libraries. If they are in combination with CIS Display, then this is exponentially increases. So if you have any questions, any project that you have in mind that you feel could be suitable, or if you want to discuss how to successfully can we drive it forward, we would love to hear from you. We have a diverse and experienced team, so just send your questions and ideas our way, and we will do our best to help you to deliver successful projects. Thank you.